All right. So let's look how these ligands bind to these receptors. All right. We have our ligand-gated channels. We have G protein coupled channels. Okay. With ligand, generally we're thinking of ionic. Okay. Ligand binds to it. Sodium, potassium, one, both. Come on in. With G protein, we're binding to a G protein. Okay. Binding to a receptor, and that activates our alpha or beta subunit of that G protein, which goes and activates cyclic AMP, right? That's the one that we talked about. And then that activates a protein, which causes magic to happen. Sometimes it opens a channel. And then we get either excitation or hyperpolarization. There, all right. So, are we going to open a ligand-gated channel? All right, so that's right here. And, there you go. Okay, we have our ligand-gated channel. We have the specific locale where it will bind with said receptor. Okay. And you can see one can dock, but these things usually bind with two molecules of acetylcholine. Okay. Again, these are, like I keep saying, an ion channel. They are ionic channels. Ionic channels, and I'll spell the word IONOTROPIC, ionotropic. Okay, these are ionotropic receptors. All right, so depending upon the textbook, all right, they'll refer to it in a couple of different ways. Ligand-gated, ionotropic means in relation to an ion. Ionotropic does. So an ionotropic type of channel just tells you that an ion is traveling through it. A ligand-gated channel is telling you basically the same thing. A ligand binds to it and an ion runs through it. For our nicotinic receptors, let's just say, all right, we have two spots to bind with for acetylcholine. When it comes to this, we can get both. We can get sodium coming in. We can get potassium going out. Generally, more sodium comes in than potassium goes out in these cases. Okay. So it does have a tendency to be more excitatory in nature because of that phenomenon. And that's what they're trying to show you here on this slide. Okay, here's your nicotinic acetylcholine receptors. And it specifically, excuse me, gosh, it specifically opens this ionic channel. And as you can see, more sodium is kind of coming in than potassium is leaving. One of those neurotransmitters, one of those acetylcholine molecules pops off and the thing shuts up. So when it comes to these nicotinic acetylcholine receptors, and the ligand is acetylcholine. Okay. So when it comes to this nicotinic acetylcholine type of receptor, I need both of these neurotransmitters to dock to get this thing to open. If only one's there, you can see from the picture, it remains closed. It remains closed. Where would I find nicotinic acetylcholine receptors? We're going to find them on the dendrites. Okay, I'm just trying to make a nerve cell body here, and I'm trying to make an axon. I'm not doing a very good job. I am not doing a very good job. Let's try that again. There we go. I guess that's a little bit better. I guess if you're 3, this is fantastic. Awesome. Okay, so where am I going to find these nicotinic receptors? I'm going to find them right there. Okay, I'm going to find them where the synapse comes and meets. Okay, so that's where you're going to find these things. You can find the nicotinic receptor right there. All right, that's where you find a lot of these. Again, when sodium rushes in, what do we know happens? It's always depolarization. It gets closer to 0. So we go from -70 and we get closer to 0. Get closer to threshold, which is -50 millivolts. If we're on the dendrite, if we are on the nerve cell body, then we are creating that excitatory postsynaptic potential. And remember, I said it usually takes more than one EPSP to ultimately cause an action potential to occur. Okay, and you can see this right here. I have a nerve coming in. Okay, here's electricity here. Comes here and opens that nicotinic acetylcholine receptor. I just wanted to say it really fast. I have another nerve. And here I have another nicotinic acetylcholine receptor. If they both come, okay, this one and then this one a split second later, you can kind of see we can get these things to add up. Okay, and get it closer to threshold. The closer these EPSPs are to one another, the faster I can fire. Now, let's just go sidebar real quick. What if this was an inhibitory? Then obviously it's going to hyperpolarize it. Okay. Yes, sodium potassium pump is going to try to get it back to threshold. All right. And as soon as it hyperpolarizes, it's and when it starts to repolarize, okay, or starts to get back to threshold, it'll come back up and another inhibitory event can come down here and it can do it again and it can inhibit that thing again. That thing, I mean nerve. Okay, so if this is an inhibitory postsynaptic potential, it's going to go down. And when something is added together, what's the fancy mathematical term that we use? Anybody? When we add?
Multiple. No.
When we add?
The sum?
Sum, yes. I was told there would be no math today. Okay. So this is, when we add them together, this is summation. Okay. So these things can summate. Is that even a word? I don't even know. These things can be added up. A-T-I-O-N. Summation. Okay. This is summation. How do I want to visualize this? If we have an excitatory postsynaptic potential come in, okay, and we get sodium rushing in, okay, and then right after it, reaches plus 30, right, then that stops in that locale. Okay. So We get to plus 30, it starts to come down a little bit, but right behind it, oh crap, on another dendrite here coming on that nerve, boom, another excitatory postsynaptic potential, just immediately right behind that, okay? As we started kind of going on the dip down here, and then more came in, so boom, I can add to that sodium within that dendrite region. And another one comes in, and I keep adding, I keep adding, I keep adding. And that's that threshold. That's that summation. Okay. The summation that occurs there. Summation. Good Lord. Summation that occurs. All right. And this is what we mean by a graded potential. Small graded increments to get it up to eventually have enough to travel across. So that's why I say it takes generally several EPSPs to generate an action potential or to be able to establish an action potential. Okay. It is that summation that allows this electricity to travel across the surface of this sucker to get to the sweet spot, that axon hillock. Because again, what do we have in huge numbers here at the axon hillock? What do we have in huge numbers at the axon hillock? Sam, shoot me.
Channels.
Which kind?
Sodium gated channels.
Because
Because potassium is different. Where are those ones located again?
You will still have some here. It's just that we have a greater number of sodium channels, sodium voltage gated channels here.
Because it's the place where it has to start in order
Yep.
for it to.
Yes. Because, I mean, I can have a potassium channel here and it's going to inhibit. I can have a potassium channel here and it inhibits. All right. But there at that axon hillock, we have our highest concentration of numbers when it comes to sodium voltage gated channels at that locale. Okay. So everybody with me here on this and this concept. Okay. The book does a great job of putting fantastic tables in there that kind of sum up everything. Okay. So when we think of an EPSP, an excitatory postsynaptic potential, you think of electricity being generated and electricity traveling. And if you're sitting there contemplating, having a drink of water, you're like, man, an action potential is the same damn thing. Electricity is generating and electricity is traveling. But remember, EPSPs may not make it. Right? A impulse may go down and it may not be enough to travel from the dendrite across the soma to the axon hillock. So it may or may not even cause the nerve to fire. But an action potential is an all or none response. With the action potential, we open up those sodium voltage gated channels and we have depolarization. True depolarization, where the thing travels the entire distance. Here, with an EPSP, I generally need a neurotransmitter. Okay, need a neurotransmitter to open a channel. With an action potential, these are sodium channels again that cause the depolarization, that cause the all or none response in the axon. With EPSPs, they're at the dendrite, they're at the soma, nerve cell body. Channel carries both. Okay. Channel carries both. Sodium rushing in, potassium rushing out. And with that, obviously, I mean, I need multiple of those EPSPs to have enough strength for that wave to surf across the dendrite, the soma. So it just loses charge, it dissipates. It's got to travel through and on the surface of that. When it comes to an action potential, I mean, the sodium channel opens and yeah, depolarization. And then we get to plus 30 millivolts and it inactivates and closes. And we repolarize. So I can send something down the axon again. Excuse me. So potassium is going to be involved. And with an EPSP, it just dissipates. Okay, it just dissipates. Conduction distance, that just tells you, you know, how far can it travel? I'm not going to ask you how many millimeters it travels. Okay. But you can see an EPSP is very short. Okay. Very short distance. Dendrite, not very long. Nerve cell body, not very big. Okay. For an action potential, we regenerate it at every node of Ranvier or at every single square inch of that unmyelinated nerve. I open up every doorway to allow this action potential to propagate down. Okay. Is it a positive feedback mechanism? Yes. The electricity causes that sodium voltage gate channel to open, and that travels a distance, and that opens up the next one, positive. And that travels a distance, opens up the next one, opens up the next one, opens up the next one, yada, 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 yada. So it is a positive feedback mechanism. EPSP opens up, and then we kick out the acetylcholine molecule. It only stays there for a little bit of time. Maximum depolarization, I don't necessarily care about. Okay. Plus 30 is all we need to worry about. As you can see with an EPSP, it gets closer to 0. Okay. And that's about it. Okay. Doesn't get to plus 30. Summation, again, it's an all or none of, event for an action potential. So there's no summation. Okay. The summation is what happens on the dendrite or on the soma. Refractory period, yes, on an axon. Okay. I have to inactivate. I have to close. I have to open. I have to reset. Okay. When it comes to an EPSP, they're both coming through the same dang hole. All right. So there's no general refractory period there. Okay. Impulse comes in, sodium comes in, a little bit of potassium goes out, tiny little wave of depolarization occurs. I can get another impulse coming down right behind it, cause it again. That'll add to the wave. And the more I add to that wave of depolarization, summation, the greater likelihood I have a chance of getting to that axon hillock. Effective drugs, we're not getting into that. I want you to understand how these things work. We're not going to get into drugs. Tetrodotoxin is just a sodium channel blocker. That's all it is. Okay. I think I have a slide that kind of shows, what some of these botulism, curare, what some of these toxins actually do to these things. So it's kind of interesting. Obviously, if I have a toxin that blocks a sodium voltage gated channel, will the nerve fire? No, right? So.
So for an axon potential, it's all or nothing. But for an EPSP, there has to be enough of these like little tiny bits of energy to add up to hit threshold. And if they don't, nothing happens. Okay.
Because when it comes to those EPSPs, Sam, they just travel a very short distance. Okay.
It's not like an action potential where it's a quote unquote longer distance,
Correct.
even though it's still relatively short.
Correct. Okay, correct.
So an EPSP is even shorter than the distance like between the nodes of round VA, for example, if
Yes.
it was
Yeah.
traveling down.
Yep.
Okay.
Yep. And look at it this way. You see this, you see this coming in right here and forming a synapse. We see this here coming here and forming a synapse. Okay. This electricity is going to travel. I mean, you drop a rock in a pool, right? Or in a pond. The ripple goes out in all directions. It just doesn't go out in that one direction, right? It goes out in all directions. So here, I have this electricity coming in and triggering receptors here. So it's going to spread across in all directions here. Okay. So what's right here? What can I trigger right there? If this thing is allowed to get to this point, can that trigger that thing to open up? Sure.
Yes.
Okay. If this impulse is coming down this nerve and it branches, is it going to open up both spots? Yes. Okay. Am I causing a voltage channel, event occurring there with an EPSP? Yes. Am I causing a voltage event here with an EPSP? Yes. Okay. So I have sodium rushing in here and then more sodium rushing in here. And that sodium adds. And that sodium adds up. So it allows that allows that wave of electricity to keep traveling. Bigger the stone, longer that travels. Bigger, more sodium
The more
coming,
sodium,
the greater that wave can travel.
bigger the ESPS.
Okay. And again, as you can kind of see, it's not uncommon to have a nerve cell body, have 50 to 100 synapses on it. It's never this idyllic thing that we think about with one axon,
Only
one
four.
dendrite, one axon, one dendrite. I mean, it is in a nerve. Okay, sure. I mean, there are scenarios where it goes 1 to 1, but there's 100 of them going 1 to 1, or 1,000 of them going 1 to 1. But when we look in the brain, I mean, good God, it is not uncommon to have 10, 50, 100 or so synapsing on one dendrite / soma of one neuron. Okay. We're thinking of it in very simplistic terms. One axon, one dendrite, one EPSP. But in reality, it takes a lot of these things. And there are a lot of these things synapsing with it. And that's hard to visualize. It's very hard to visualize. So I get it. Okay, let me do this.